graciously chair and moderate the session. Dr. Karyakarte sir boasts over 35 years of exemplary experience in the field of microbiology. Throughout his illustrious career, he has held numerous academic and administrative positions, ranging from assistant professor to dean, leaving an indelible mark at every level. Sir's contribution has been recognized with prestigious awards, such as the Best Medical Teacher Award by DNR, the COVID Yodha Award by Deputy Chief Minister of Maharashtra, and the Dr. Pratibha Nayak BAMN Award by the Vinod Association of Medical Microbiologists. His dedication to education and research is evident in his mentorship of 15 postgraduate students, with publication of over 120 research papers in reputed journals, and leadership of 17 research projects in collaboration with national and international institutions. He penned the MUHS approved textbook of medical parasitology. During the COVID-19 pandemic, his efforts were particularly noteworthy. He collaborated with esteemed organizations such as IGIB New Delhi, Pune Consortium of Genomics, Maharashtra Consortium for Genomics, India, and ESET. He serves as a member of the Maharashtra State Task Force for COVID-19, the State Coordinator for Genome Sequencing, and the Coordinator for statewide in all government medical colleges. He was the divisional RTP state Pune. Sir has significantly contributed to IIM as well, serving as President and Organizing Secretary of Maharashtra Chapter twice, and as the Organizing Secretary for Microbiology 2024. He has also promoted public awareness through over 50 radio shows, TV programs, and websites. Given his unparalleled expertise and numerous accolades, it is not possible to fully encapsulate his achievements in a brief introduction. I have the honor of handing over the session to Dr. Karyakarte sir. So please.
Good afternoon all. We are really at the risk of the post-prandial session, and I want all of your attention here because we are going to discuss something which is very important, and that is how genomics can help in an action which can be taken, particularly in relation with the pandemic control. So that is what I'm going to just elaborate. I will just be very fast. I want my other colleagues to talk more and just coordinating the whole thing and trying to place the things in perspective of why this particular session was kept. This session was mainly kept because I wanted to showcase how genomics really helps in controlling a pandemic and can make the government making informed decisions based on this particular data which you get from genomics. So with this, I would like to start. The whole thing started here. It was a government resolution from the Maharashtra government which recognized that BJ Medical College had found a new mutant. And so based on that, they came up with a GR which helped us, that is Government of Maharashtra, collaborate with CSIR-IGIB to have a program which was the second program after Kerala for genome sequencing on its own, the state doing it with the collaboration. INSACOG was already there. I want to make that particular mention. INSACOG was there, but still we had a different state effort which was taken up by Government of Maharashtra. And therefore this particular session is very important because people who just made it possible are sitting right in this hall right now. That is what I wanted to introduce to you. That a big government program, all the members are sitting here right in the hall. That is the thing which was to be emphasized. So we move next. So this was how we did it. We did it in 2 sessions, one of 3 months and another 6 months. So total 9 months was the study which was there between CSIR-IGIB and Government of Maharashtra. And what it helped it was during the late phase of Delta and the early phase of Omicron that the whole thing happened. And it laid a strong foundation about control of SARS-CoV-2, that's COVID-19, telling us that we have variants which come up and these variants lead to a wave. That idea wasn't there to begin with. The new mutations which were caught, the new variant which was caught, its importance was not well understood by many, and therefore this study led us to understand that, well, if you have a new mutant, if you have a new variation, if you have immune escape, then you have a chance of having one wave after the other. So here we tested a lot of samples, as it's clear, and 18,000 in all. That was the thing which we got from the study, 18,000 samples. I will just move very fast. I'm not going to take more than 10 minutes for that. I just wanted to see and have you focus on the slides now, and you are going to get a colored view of how this particular thing changed in COVID. I will be very fast with it. See, if you look at this particular colors here, I have colored Delta with green, and you'd say that in May 2021, the domination was with Delta. So we start month-wise now. Next month we see that something else is happening. You have Delta, which has added mutations, and then they classified it as AY.1, AY.2 and all. So you had a change. If you see the colors, you would appreciate this. All are, the single bar stands for districts in Maharashtra. So all the districts are covered here, and you see that there is a pattern which is there all over the state of Maharashtra. So you can understand the speed with which these variants spread at that time, and still now also. That in a month, you are getting a change in the variant all across Maharashtra. So to begin with, it was Kappa, and in no matter of time, just a month, you are having domination with the AY series. As we move further, you had the introduction of Omicron for the first time in December in India. So this is a dramatic slide of mine where I try to emphasize of how these variants have emerged and how fast they spread. So in December, you see traces of Omicron. You see traces of it, still dominated by Delta and the AY series, but you see what happens in January. The picture just changes. So in 1 month, Omicron has replaced the entire population of Delta. So that is the speed and scale with which this pandemic evolved, and our study shows it very clearly of what happened in the pandemic. This is what happened, and therefore you had waves after waves after waves. Right now, so we move further. So this is how I just wanted to focus on Amravati, because Amravati was a very important focus during the pandemic. Sarukhe sir, he went to every village there to see what happens to the patient at his age. He traveled everywhere and he saw what was the problem, and then advised the government of Maharashtra as well as government of India that you have to act fast. Government of Maharashtra responded with lockdown of Amravati, and that led to saving of many, many lives. So that is what happened. And if you see Amravati at a close, you would understand how the variations spread. And first it was Delta, then AY, and then lastly it was totally dominated by Omicron. There were two very important situations during the pandemic where I want to emphasize of how genomics can change your outlook and how government decisions can be taken. Now let me tell you, you have to remember that particular time when you had a second wave and the third wave was to happen, and everybody was saying that it is going to be in the pediatric population. And therefore government took a decision of having many NICUs, PICUs, invested heavily in it, and then what happened was, suddenly you had a lot many children, 50 odd, affected in Aurangabad, Chhatrapati Sambhaji Nagar. So what happened was, there was a panic that a third wave has started now, and it's going to affect on the effect of the children. So what we did was, it was really jet speed. We could get the samples from Aurangabad and we sent it to IGIB. And there they had devised a new method. The new method was a faster way of doing sequencing with the help of Willow. That led to this fast sequencing was then adopted all over the world, and it's credited to CSIR-IGIB. That particular set of Illumina protocol was established. So with this, we sent these particular samples to New Delhi with CSIR-IGIB, and within 48 hours, we got the results. And the results are in front of you. What does it show? It shows that it was delta predominant, which meant that there was no new variant, which also meant that there was no need to panic. There was no chance of having pediatric infections. So that led to a decision by the government where a panic button wasn't pressed, lockdown was not reintroduced, nothing happened. So that particular thing, a decision which can cost the exchequer billions and billions, was not taken because the lockdown is worth many, many thousands of crores. So that decision has to be based on something which is very, very pertinent, which is very, very important, and that important thing was knowing the genomics and knowing the variant. Same thing happened. Now this was, I think, Vinod would remember this. He found one particular mutation there, and that mutation was very important. That was the K417N. And he alerted us that this new mutation can be a dangerous mutation, it can be something which can cause trouble. And we went on very fast. It was found in, if I remember correctly, it was in Ratnagiri. And from there, now here I would like to emphasize how the whole administration can jump in. I had a phone call to the Secretary of Medical Education that something, I was alerted by CSIR-IGIB that there is something new. So he said, what should be done? I said, we'll take at least 50 samples from that particular patient and his relatives around him, and then his close contacts, and let us see what is there, if anything new is there. We did that thing again. Again, 50 samples, in 48 hours, we got the results, and the results told us that no, it was Delta which was predominating, and the new variant was not holding ground. That is what we got. And further, there was no increase in the basic clinical pathogens level which was there. So that we went forward. Similar incidences took place. We did what we can do, and that was only 48 hours. We could get the results. So that was very, very fast genomic diagnosis which we went about. All these things tell you how fast it was. But then most important thing which happened with us was, INSACOG, we then had an entry in INSACOG. Because up till now we were outsiders. But then we were doing so much of work that INSACOG had to take us. And Sir was there. They all saw to it that we come in INSACOG. And then INSACOG was expanded from 10 to many other. So we sought to be attached with NCCs, with DBT. That is how we went about the event with DBT. And then later on, INSACOG, once we went inside INSACOG told us that this is not the only thing. We can't just have the sequencing done and no other thing for following it. So we did clinical studies. We went on and asked questions telephonically to all people of what were the symptoms and signs. With that, we got the clinical study and the clinical presentations, number of admissions and all. This helped us and tell the government that there is no increase in pathogenicity. The thing is spreading fast, but it's not causing many more deaths than it is causing right now. So this went on and on with each variant. BJ Medical College did the clinical study. So that was the power of what could be achieved by a single MOU, a single decision of the government to strike at a pandemic with the help of the latest technology, and that is sequencing. So I won't go further now. I would give because I think I have made a platform showed you a platform of what happened actually, and then I would ask each individual member to tell us what was the role that was played. I also have Ravi with me, who will be telling about the clinical side of the whole thing, which would be some other than the things which we are discussing. I also have Rajesh Pandey here, who would be telling about what innovative he did, because he is known for sequencing at the airport. So that is it. So I think with this I will stop. I won't be going ahead with my further slides. I will stop it. Thank you.
Thank you, sir, for a comprehensive overview about the session and setting the stage for our speakers. Our next speaker is Dr. Vinod Scaria. Dr. Scaria, sir, is a clinician turned computational biologist. His research spans the application of genomics and informatics in clinical and biomedical settings. He is a medical researcher, pioneering in precision medicine and clinical genomics in India. He is best known for sequencing the first Indian genome. He was also instrumental in sequencing of the first Sri Lankan genome, analysis of first Malaysian genome sequencing, and analysis of wild-type strain of zebrafish and indigene program. He is the co-founder of Genomics for Understanding Rare Disease, India Alliance Network, popularly known as the Guardian. He has been a part of collaborative genomic projects. He has also been a part of early whole genome sequencing population genomic projects, including the Indian, Sri Lankan, Malaysian genome projects, and is a member of the Hugo Pan-Asian Population Genomics Initiative Task Force. The COVID-19 pandemic has seen his lab pioneering in genetic epidemiology of SARS-CoV-2 in India. He spearheaded the initial state-level genomic surveillance programs in India, and his research contributed to the earliest reports and significant understanding on immune escape variants in COVID-19 reinfections and vaccine breakthrough infections. He has over 200 research publications in international peer-reviewed journals and 2 books to his credit. He was previously associated with CSIR Institute of Genomics and Integrative Biology as a senior scientist, and is presently working as a senior consultant at Karkinos Healthcare and an adjunct professor at IIT Compere.
A very good afternoon to you all, and thanks Professor Karyakarte for inviting me here. And as usual, Professor Karyakarte, being a professor in microbiology, asks very difficult questions, and he has not spared me this time also. He asked me to talk about pandemic preparedness and what I think should be the pandemic preparedness plan. So I thought I can sort of summarize the multiple interactions we had over the many years during the COVID pandemic into trying to understand what should we really do in terms of the pandemic preparedness. So next 15 minutes, that's what I'll try to summarize. Now why is this important? It is important because of the fact that we do have infections happening across us. To put numbers, there are about 200 odd estimated outbreaks every year with a potential pandemic potential happening across the world. And even if you look at India, there are many such active outbreaks that happen, many of them seasonal, some of them new outbreaks. Zika, Nipah, so on and so forth are just names that you can add to the list. But the most important thing is that many of these emerging infectious diseases do not have very well approved diagnostics in the market, which is widely accessible and affordable. And that necessarily would mean that very few centers will be able to do the diagnosis. And that is something that we put into perspective when we talk about pandemic preparedness. Now, in many ways, our ability to look at pandemics and ability to understand the pandemic potential of pathogens have changed quite significantly. And some of this is thanks to the COVID pandemic. And the first of it is largely that the world has transcended to a digital ecosystem. That means that a lot of significant data is available in a digital format that necessarily the computers can be put to work to pass through this genomic or rather epidemiological information which is already available in many digital formats. The second is the mainstreaming of genomic applications, not just in SARS-CoV-2, but also increasingly in looking at many other pathogens, and I'm sure the other speakers in this session will talk about many such applications for many of the emerging pathogens. And the third most important is openness as a virtue. I mean, before pandemic, practically everybody would think that I have a genome and I keep it in my pocket and probably nobody would ever look at it. That has largely changed with COVID because of the fact that, I mean, unless you really put it out there in open, it's really not going to benefit anybody, including yourself. And that openness has actually changed the way we look at genomics and we look at outbreaks and genomes from these outbreaks. Now, in my thought, there are largely three things that would entail to a preparedness in terms of the pandemic. The first is our predictions and early risk assessment. If you find a pathogen or if you found an outbreak, can we sort of get an evidence-based overview of is it likely to become a pandemic? The second is how can we really put this insights and evidence into building policy, because early policy and early intervention is something that will change the course of the pandemic. And last but not the least, but probably the most important one, is the rapid approaches for diagnosis, treatment, and surveillance of the pathogen. And I'm sure you've heard about Dr. Karyakarte's work in our ability to look at sequences very rapidly from emerging outbreaks across Maharashtra, but this could now be extrapolated to many more outbreaks, not just SARS-CoV-2, but many other pathogens happening across the world and across the country. Now, in India, largely, I think there are three things that we lack. And the first is an epidemic intelligence system. I'll come to what an epidemic intelligence system is. Second is, how do we connect these dots, right, from the epidemiological intelligence system, which will gather information from across the world and across the country, and put this into an epidemiological perspective in terms of making evidence-based assessment, which is extremely important in policy building. And of course, the last is the rapid assessment and early action. And in most epidemics which has happened even after the COVID pandemic, you would see that this is something that is largely lacking. And if you can put these three things together, I think we can do a quite tremendous job in our ability to predict pandemic potential pathogens and also be able to sort of contain them very early on. Now, this is one example where we put together, and this largely came with a set of volunteers, around 500 odd volunteers based out of Kerala, who came together to create a very cohesive group, what we call as Epidemic Watch. Now, Epidemic Watch is essentially a volunteer group which includes doctors, epidemiologists, bioinformaticians, researchers, and of course lay people to keep a very constant track of outbreaks that happen not just in the country but also across the world. And there's a elaborate way of discussing this out in terms of what is likely to be a pathogenic, an outbreak with a pandemic potential, and in many ways track it to a sort of granularity that we have almost daily updates on every outbreak that happens across the world. Now, the second is, of course, digital ways of looking at outbreaks. And you will see that many of the countries have actually moved into a surveillance system which sort of asks its participants or volunteers on a very constant basis about outbreaks that happen. This includes the very famous flu survey, which is done in the UK, infected radar, which is done in Belgium, and so on and so forth. There are many such initiatives across the world. And infected radar in India is one such initiative which is based in Kerala, run by the Epidemic Watch Group. Now, how do you really put this together? And to put this together, you need to have multiple different sources of information. One source of information is, of course, coming from the global context in terms of emerging data from outbreaks across the world. For example, if you look at the H5N1 outbreak in the US, in the cattle, are there people able to sort of put it in a granular fashion, compile it so that we can sort of understand when is it going to, or when is it likely to come to an Indian context? For that, we need to pass news, and of course, we have automated tools today in open source which are built as part of this program to be able to track. And of course, the second important part is human mobility and mobility networks. This is very much important in a state like Kerala, which has almost 3.5 million people who are expatriates who are spread across the global diaspora. That would necessarily mean that any pathogen anywhere in the world, probably the first state to be affected would be the state of Kerala. And of course, the second part of it is the local or the regional insights, which come from lay people who are constantly tracking outbreaks, even very simple symptoms like fever in their local hospitals to give early alerts to say something really is happening abnormal in this particular location. And put together, there's a team of people who do a sort of risk assessment, putting all this information together and trying to make very useful guesses. May not be scientifically accurate, but nevertheless very importantly useful in terms of looking at what needs to be made with that taking in. Now the other point that I want to make is that many of the data sets that come from not so related fields could be used to build such evidence-based models. And just to give an example of the dengue outbreak in the state of Kerala, the volunteer group had sort of mapped out every single dengue case in 2021 to create a sort of a map of dengue across the state to understand what are the hotspots of dengue. Now that map has come in very useful in our ascertainment of where is Zika going to likely happen in the state of Kerala, because of the shared vector. You would necessarily mean that the hotspots of dengue would also eventually serve as a hotspots for dengue, for Zika. And this can necessarily become very useful for policymakers because the countermeasures could be implemented very, very in advance because we essentially know where the mosquito population or the disease is likely to happen or emerge in the future. And another such data is essentially very useful for tracking the H5N1 outbreak. The group has been tracking H5N1 outbreaks for now 4.5 years. And this comes from a very basic principle of our ability to track every outbreak across the world and put it in the context of migratory birds, or what we call as the flyways of migratory birds across the world. And pretty much with, I would say almost 4 weeks accuracy, the group can pretty much predict when the outbreaks are going to happen in the state of Kerala. We have done this consistently for the last 2 years. And just to highlight the point that many information that might not be directly related to the disease, but could actually be pulled out from known facts from across the world, could actually be put together to make very useful guesses on when an outbreak is likely to happen, especially in outbreaks like this. The second part is about our ability to sequence or use genomic sequencing to understand emerging outbreaks much better. And this is even more important given the context as in the past. Every outbreak would take probably weeks, if not months, in some cases even years, before the pathogen was diagnosed, because it followed a traditional process of microbiology which involved culture, isolation, and observation of the organism. Now, genomics is something that has sort of sidestepped this altogether, because you can offer a very quick, efficient way to diagnose an outbreak even before the traditional microbiology processes have set in. Because the traditional microbiology processes would also require biocontainment facilities, would also require advanced equipment to be able to understand the pathogen. But genomic sequencing can be done on benchtop sequencers today. I'm sure you have heard about the new benchtop sequencers that are being launched, and many more are in the anvil. You could sidestep this altogether because practically every pathogen on Earth has DNA or RNA as its genetic material. And this is sort of exemplified in the SARS-CoV-2 pandemic, where within weeks' time, the Chinese counterparts in Wuhan were able to sequence, annotate, and also be able to characterize in the epidemiological context in terms of an outbreak that's emerging with a new variant of the pathogen. So genomics offers not just detection or characterization of the pathogen, but also its ability to put in the evolutionary context in a phylogenetic way. And there are now many methods to look at in an unbiased way pathogens that can come from clinical samples. And this is one such approach that we sort of put out way back in 2011 in our ability to call whole genome sequencing. And this essentially sequences the entire clinical material, and using advanced computational tools, you would be able to sort of isolate out reads that come from a pathogen without even knowing what the pathogen is. You you can make very useful assessments of whether it's a virus or bacteria or a parasite because of the genome sizes and the context differences of these genomic reads, and be able to probably also assemble genomes in probably its entirety. We have many such examples from human as well as animal pathogens using this particular technique. And the third application is our ability to do this at scale, and I'm sure you have heard it from Dr. Karyakarte on how large-scale genomics can happen in our ability to surveil the pathogen over time course. And this largely happened with the COVID-seq assay brought together by Willow, and we were one of the first labs in the world to be able to implement in a public health setting, thanks to grants from CSIR, our former Director General is in the audience. And this technique essentially allowed us to sequence 750 to around 3,000 samples in less than a day. And that's the kind of throughput that would be required to do large-scale surveillance programs or state-wide surveillance programs that you would heard about in the previous talk. Just to give an example, Kerala was one of the first states to be able to do such a surveillance program very well before in summer. And over time, the surveillance program in a systematic way, were able to trace very emerging pathogens, variants of the pathogen across time courses. And on the top, I've also sort of mentioned the public health measures that were taken based on the insights from genomic sequencing. And this includes the double masking, the statewide lockdowns, the active tracing of the Delta Plus variants, and of course the surveillance of Omicron linked to 2021. And this is possible not just because of the ability to sequence very fast, that's very important of course, but also the ability to analyze the trend in real time or near real time, and that's only possible if you can automate the entire process of analysis, interpretation and reporting. And one such tool that's widely used across the world, at least in the WHO, is CRO region, is a tool that was developed in a lab called the GenePy box, which was built by Dr. Bani, which now automates the entire process of analysis and interpretation, and also the reporting in a public health context, so that the reports can readily be shared with the public health personnel. Now, the other important part of doing such surveillance programs is our ability to make diagnostics much better. And in fact, the SARS-CoV-2 pandemic has been a good example of how we could do this. By looking at regions in the genome which are targeted by the RT-PCR primers and looking for genetic mutations which can change the primer binding site, or rather the efficacy of the primer binding sites, you could now sort of go back and build better tools to be able to look at either emerging variants very specifically, or look at sort of future-proof primers which can be used across the virus genome. And we have at least one such kit which is now widely marketed by Siemens, and it was developed in IGIB by looking at thousands of genome sequencing, and to also identify regions which are evolutionarily conserved across the genomes of these viruses, and be able to optimize the diagnostic procedure by looking at genomes. Now, before I close my talk, I think what is really more important is collaboration across the spectrum. I mean, it's not just Professor Karyakarte collaborated with us and this just magically happened, but it was just because a large ecosystem of the state healthcare machinery was part and parcel of the collaborative efforts. And I'm sure you will cherish long interactions which we had with not just the Chief Minister of the state, but also the public health personnel who were managing each and every district as part of this program. So it's not just education institutions or research organizations, but clinical public health research centers, the state and the local governments, and of course volunteers who do a lot of the weightlifting which makes this ecosystem possible. Thank you very much. And just to take a few take-home points is that epidemic intelligence is what is required along with genomic surveillance for pandemic preparedness. And genomic surveillance can enable early insights and examples in the previous talk, and you will hear more in the future talks. Evidence for diagnostic development can be accelerated if you have genome sequences very early on, and by doing a surveillance program, you can optimize the diagnostic development very efficiently later in the process. But this is all possible only if you collaborate quite efficiently and extensively. Thank you very much.
moving forward, I call upon our next speaker, Dr. Rajesh Pandey. Dr. Rajesh Pandey, sir, is a principal scientist at CSIR Institute of Genomics and Integrative Biology and an associate professor at the Academy of Scientific and Innovative Research in Delhi, India. His expertise and research interests include integrative genomics of host-pathogen interactions, functional genomics, non-coding RNA biology, single-cell genomics, transcriptionally active microbes, and innovative, cost-effective applications of NGS technologies. After his PhD in biotechnology, he worked as a research scientist at MRC Harvard Institute in Oxfordshire, UK, and as a visiting scientist at Johns Hopkins University in Maryland. He has published 86 papers in reputable international journals with a cumulative impact factor of exceeding more than 560. We welcome you.
When I received the itinerary, I saw two people will be speaking or preceding me, Dr. Karyakarte and Vinod. So I thought, well, what do I speak which doesn't overlap and at the same time provide something new to the participants over here. So some of the slides which is there, I may be going fast. Some I may spend a bit more time just to bring a new perspective to how pathogen genomic surveillance would be strengthened going forward. So, during this journey of last 5 years or so, in terms of we all being exposed to the challenge of SARS-CoV-2 or COVID-19, what we see here that when we see differential disease severity in society, in hospitals, we have to take a step back and see that, well, how much of that disease severity which we see can be explained by which of the different factors. So what I have tried to put together is some of the factors which we have worked in the last few years to, and those have been quite useful in terms of understanding that this all contributes to differential disease severity, which includes the pathogen causing the disease, differential host response, then what is the role of resident microbes within those individuals in which infection happens. Then if we go a bit deeper and look into how RNA plays a role in that in terms of one gene, multiple transcripts, and then digging deeper, if a virus enters into a cell and if we look into a single cell, do we find any remnants of those microbes within that single cell, and whether that is any useful to help us understand the differential disease severity. These all are interconnected, and that all has been developed in the form of a program. We call it as integrative genomics of host pathogen, which synonym as IngenHope. We are hopeful that some of the research we are doing would be useful to the community going forward. Okay, so these slides I will be skipping, as I said, because this will be a repetition of some of the work which we have already talked about. But I will spend a couple of minutes on this slide. So when we looked at different aspects, we found that when in our collaborative effort, we found that a group of patients when admitted to a hospital, and they were diagnosed as lower respiratory tract infections, and we found that some of them develops pneumonia and some of them doesn't have pneumonia. So we asked a question that, well, what may be the role of different microbes coming together within it, and whether or not resistance towards the treatment with the usage of antibiotics may have any role. So we again use the strength of genomics to look in an integrative way where we queried for bacteria, viruses, fungi, and AMR alleles at one go, and we found that the group of patients who had pneumonia were actually given class of antibiotics for which they already had resistance by virtue of the presence of those microbes within them. whereas the group of patients who didn't have pneumonia were actually didn't carry those resistance genes. So now, going forward, imagine a situation where when we develop an SOP wherein a clinician has an advantage of knowing the potential AMR resistance in those patients and whether that can help clinician to take a decision in terms of which class of antibiotics which may be used. So that's the aspect which we will be digging deeper. But if I summarize a whole lot of classical pathogen work which has been happening in the lab, so these are the diverse aspects of work which happened in the lab. What you see on my right hand side or your left is the first samples which came to IGIB for sequencing of 2 samples of SARS-CoV-2 to the one which happens on that front of it. That's the mobile sequencing which happened at the Delhi airport for knowing that international travelers who came to India during 2 different waves of Alpha and Omicron, how many of them may be carrying Alpha or Omicron based on RT-PCR at the airport. They were sequenced at the airport itself, and then those reports were shared with the government to decide that whether they will be hospital quarantine or they can also have a home quarantine as well. So this summarizes a combination of clinical genomic surveillance, community genomic surveillance, and how those insights have been useful to help take public health decisions. And those insights which we had developed during the time of SARS-CoV-2 has been extremely useful going forward for the last 3 years of dengue genomic surveillance which we do. And again, the work which happens is towards why a group of individuals infected by same or similar pathogens has dissimilar disease severity. So the whole aspect of looking at the pathogen, looking at the host response, looking at the resident microbe, and looking at the potential presence of microbes within the single cell are also being investigated in terms of dengue and its causing of differential disease severity. So one of the ways in which, and to this audience, it is also important to possibly bring to the fore that how or what are the strengths of genomics as well. So one of the things which I think I would be happy to highlight is about how do we make multiple use of the same genomics data. For example, what we do quite in depth in the lab, that if we have done, say for example, total RNA sequencing of a group of patients vis-a-vis clinical data collected, can we use the same RNA-seq data for answering multiple questions? And that's actually feasible. How it is feasible? So based on different group of diseases which we have worked on from COVID-19 to dengue, we realized that between 60 to 70% of those RNA-seq reads map to humans. Remaining around 30% of the reads don't map to humans. Initially we thought that what may be the cause? Is it like we need to improve anything on the library preparation or data analysis, and we realized that that's not the case. The leftover data of non-human reads are actually a wealth house of information which can be used to strengthen this genomic surveillance and strengthen this aspect of integrating genomic surveillance. So if you are working on an RNA virus disease, then RNA-seq data can be used to understand viral pathogen genomes. You can use the data classically, of course, to know the host response in terms of coding and non-coding. At the same time, you can use the data to understand the role of long coding RNAs in it. And as we all know that as we stand here and we are also resident to a lot of other microbes within us, which at the moment are not causing any disease per se. But if a new infection happens to us, what happens to the dysbiosis of those, and whether that dysbiosis leads to any disease? And what we found that perturbations in those transcriptionally active microbes, which is captured through RNA-seq, is a very interesting toolkit to understand that how they are related to disease severity. The same approach we have now translated to single-cell transcriptomics data as well, that if you look at RNA-seq data within one cell and then you ask these questions, that will, in addition to, if it is RNA virus, then in addition to RNA virus, if you look at host response, coding, non-coding, and as well as looking into the remnants of potential microbes present within those, can we answer all those questions? So this is the broad framework on it, and now we'll give 2 specific examples that how this idea has been useful to answer some of the questions. So when SARS-CoV-2 happened initially, we worked through different phases of SARS-CoV-2, which has been named as different cohorts of it. But broadly, when we initially had an understanding of SARS-CoV-2, we saw different disease severity and based on the clinical partnerships and the clinical data for those hospital-admitted patients, we identified as mild, moderate, severe, and unfortunate mortalities as well. And we asked a question that what is difference in this, that although they are infected by same variant of SARS-CoV-2, why they are having dissimilar disease severity? So in addition to other aspects of the work which happens in the lab in terms of looking into pathogen genome or host response, in one of the aspects of the work which we looked into, that what is the role of those resident bacteria which is present in those individuals when the infection happened and whether they are contributing to any observed disease severity. So for that, what did we do? We took samples, we made total RNA-seq libraries, we did alignment of those reads with the human genome, and then we were left with some non-human reads, and then we took those non-human reads to look where do they match to? Are those mapping to bacteria, fungi, or viruses? And we found that a good subset of them were mapping to transcriptionally active microbes. Please note these terms, transcriptionally active microbes. This is a variant of functional metagenomics in our understanding. And then we looked at those dynamics in the different disease severity phenotypes which we were looking for. And to take home the message forward, what we looked at, that when we looked at the alpha diversity of these bacteria within those mild, moderate, severe mortality patients, we didn't find much difference. But when we looked at the beta diversity, we did start to find the difference in terms of abundance of those bacteria. So summarizing the long story, what was the take-home message? That we found 6 different bacteria which were different between mild, moderate, severe, and mortality. But how did we reach those places? So if you look here, what did we see? If we look at non-human reads, we find that the presence of non-human reads, in other words, bacterial reads, is more in the severe and mortality patients. Then we said, okay, which of these are those? Please remember that these are kind of subset of the RNA-seq reads. So we were very stringent in terms of what cutoffs do we apply so that the inferences we make are resilient enough or are robust enough. So we looked at the top 30 bacteria present here, and we found that there are 3 of them which were consistent across the groups we're looking for, but then there were 6 which were differentially present in this. So this was the first of the SARS-CoV-2 work in this direction, where we identified, for example, Achromobacter psilocyclins and Bacillus cereus were highly abundant in the patients who did not survive. In other words, the mortality patients. And then going forward, what we found was that Leptotrichia vocalis was higher in the patients. Patients were moderate and severe, and then going forward, those were other ones like Leptotrichia vocalis and Velonella parvula. Then in the mild group, there was an enrichment for Actinomyces myceri and then Velomonas species. This was the first observations at that point of time, and we thought how much relevant it is. And at that point of time, this is like 3 years back, there was one study which showed that Velonella species is actually a signature for SARS-CoV-2 infections. That gave us confidence that probably it's in the right direction. Then we looked back into the literature and we found that a lot of these bacteria were actually known to have that specific role. When we go forward and try to see that why all of a sudden Achromobacter is so high in the patients who didn't survive, and it was not so difficult to explain it because Achromobacter psilocyclins was known to have a lot of resistance to the antibiotics, and possibly we postulated that because of the high abundance of Achromobacter, antibiotic usage in these patients who control for secondary infections did not work as desired in compared to the mild, moderate, and severe ones. But this was one of the initial studies in this. Then going forward, we did other studies with different severities. For example, vaccinated patients with mild severity compared with non-vaccinated infected patients, and findings vis-a-vis Bacillus cereus and Acinetobacter zygosoxylins stood true in terms of their association with disease severity. Then we took a step back and said, well, if we go forward and see what are the bacterial pathways which are enriched in this, and then what we found interestingly that in the patients who had severe or mortality phenotype, pathways related to ribosomes, bacterial motility proteins, and bacterial chemotaxis were extremely enriched. So these analyses are based on all the bacterial genes which were expressed in those patients which we did. Then we took forward, and then at that point of time we started single cell based work in the lab, and then and this is a summary of different work in this direction, that how co-presence or transcriptionally active microbes may be playing a role in affecting disease severity, and how going forward this can also be looked at in terms of development of diagnostic kits vis-a-vis pre-information towards a patient going into a potential severity. Please remember one point, that the patient samples were collected on the day they were admitted in the hospital. So this has a high potential of early markers for potential disease severity as well. And disease classification was based on the patient's journey in the hospital. So these markers can have a high potential of it. And we got more confidence with downstream studies, not only from SARS-CoV-2, but another RNA virus of dengue last 3 years of work in the lab. Now going forward, we'll spend 5 minutes on this before I end the talk is, we hypothesized in the lab that if a virus enters into a cell, and for it to multiply, it uses the cell machinery, which a lot of papers talked about it. As a genomics lab, we hypothesized that if a virus or a bacteria needs to be deactivated or needs to be killed within a cell, can we also capture the remnants of those bacteria or viruses within that single cell, and whether that can be useful marker for knowing the differential immune functions of the cell going forward. This was a hypothesis 2 years back, but now we have completed that study, and going forward we still are the only one which showed that how single cell transcriptomic data can be used to look at the presence of microbes within a specific immune cell type, and how it may affect the function of it, how we do not know at this point of time. Cutting short, we did single cell transcriptomics of 3 groups of patients: healthy patient, healthy people, active infection, and recovered individuals, and this recovered were within 1 month of SARS-CoV-2 infection. What we did find again was that in a normal line of analysis for single cell, we did find 12 different cell types. But what is important was, if you see those circles in the box in the middle, that only 82% of those single cell transcriptome data mapped to the humans, around 18% across healthy, active infection or recovered did not map to the humans. So we again looked into those remaining 18% of the data to look at that what are these 18% of the data, whereas it is supposed to be, because it is coming from within a cell, it is supposed to map to humans. And what did we found interesting was that if you see the first one that we did analysis for it, found that for every 1,000 sequencing rates, what is the percentage of microbial leads? And what we found here that from healthy to active infection to recovered, there is an increase in terms of the microbial leads present in those. Let's go a bit deeper to see that how does it stand. And what we found was that because this was a study done with a data set generated on BD Rhapsody, and then there was no other reference data at that point of time, we had to optimize a lot of analytical steps in terms of how do we ensure that these findings are robust enough. So we fixed a criteria that okay, we will take any bacterial read to be taken forward for analysis if it has minimum 5 sequencing reads in at least 10% of the cells in that category. With that criteria, we found that there were 76 bacterial species across 15 genera. Going forward, we captured total of 97,000 single cells, of which nearly 1/4, around 26,000 cells, had microbial reads within those cells. Then we looked into that, okay, if there are around 1/4 of the total cells have microbial reads. Is there any pattern here? And what you see at that corner of it, that although healthy had less number of microbial reads, but there were more cells which had microbial reads. So that's what we see there, that healthy positive and recovered had a decreasing pattern of microbial reads, although there were less number of cells in healthy compared to other ones, but there were more microbial reads in healthy compared to active infection and recovered. And we started to find that between healthy and recovered there was differences coming up, especially in the beta diversity. We find differences in terms of microbial reads. So going forward, what do we take? So we again made a bit stringent criteria that do we find any patterns in those bacteria which are present in these single cells, and we find that if we look at the healthy active infection and recovered, we find that there is distinct clusters of bacteria present. Of course not exclusive, but there is shared something, but then still we find active clusters on it. Then we looked specifically that does it make sense for the presence of those bacteria into healthy infection and recovered, and we found some of the species like Streptomyces and others which did make sense of their presence in their respective categories. But what was more important, these are different in different groups. Earlier was for being healthy, this was in positive. But what is more important for us was to look into that whether this is anything to do with the differential presence of specific cell types. When we normalized for it, and we found that there are some particular immune cell types in which these microbial reads were enriched. So then we went forward and said that, well, is it a possibility that these microbial reads being present in specific immune cell types may be affecting the immune function of those ones? So then we start to look into specifically top abundant bacteria in specific cell types, and what we find that there were species like Scaria, canis and stuff which are present in specific cell types. But the challenge at that point of time was that this was the first single cell work in that time. So we thought, okay, we will look into an independent data set for validation as well. So we found out a data set wherein they had done bulk RNA-seq but not bulk RNA-seq, and we had done single cell RNA-seq. But the difference, but other parameters were similar. So both were using PBMCs, both were using Illumina sequencing, and we wanted to same sequencing platforms. We wanted to see whether the species which we captured is also represented over there. And what we found interestingly that there was an overlap of 8 species between our study and that study, and more importantly, the ones which we found enriched in our categories of patients were also enriched in the similar category of patients. Why it was important? Because as this was some sort of a benchmarking for how single cell data can also be used for discovering specific microbes, and whether those microbes in specific immune cell types can have a role in modulating the immune functions of those, and whether they can have a role in the differential disease severity. So that's a work which is now in the public domain. Going forward, we have used this single cell data for many different aspects of pathogen genomics, which includes, in addition to the classical immune functions of T cell, can T cell also do other functions? We showed yes, it has an active role in metal ion response, LNC RNA perspective, as well as looking into that. So with that, I will stop here. But the take-home message is that in addition to the pathogen genome, which carries a lot of information, there are other factors to it which we can bring it together to help us understand why same or similar pathogen leads to differential disease severity, wherein going forward, as Vinod also highlighted, that how new technologies, new platforms would be useful to strengthen our understanding, and genomics can be co-opted in that understanding, and single cell transcriptomics is one way going forward, and how microbes within a single cell can also be a surrogate for disease severity going forward. So with that, thank you so much for providing the platform and sharing some of the work from the group which we do. Thank you so much.
Thank you, sir. Our next speaker, Dr. Vinod. Sir is a former senior professor and head department of Microbiology at NIMHANS, Bengaluru. He was also the past president of IMS and is currently serving as the head research and development at Tata Medical and Diagnostics, Bangalore.
Good afternoon, and let me thank Dr. Rajesh Karyakarte for including me in this very elite panel of distinguished scientists. My talk will focus on more on the diagnostic side and how we have exploited genomics, and of course I'll also share some epidemiology lessons we learned with genomic sequencing. So the next 10 minutes or so, I will be talking on the history of genomic sequencing for pathogen detection. I think both Dr. Vinod and Dr. Pandey have touched on it, and our experience with acute encephalitis syndrome and SARS-CoV-2. The first virus that was identified historically in human virus was hepatitis C virus, way back in 1989. Those days, the hepatitis was classified as hepatitis A, hepatitis B, and non-A, non-B hepatitis viruses. This virus was discovered by a very laborious process of sequencing, but a very smart way of doing it. They took the plasma when it was negative for A and B, did extraction, and did random priming both for DNA and RNA viruses. Got a set of sequences, assembled them, and then found out that it's a novel virus and they designated it hepatitis C. So historically, this was one of the first viruses. This is Joe DeRisi, one of the top leaders in metagenomic sequencing globally. He heads the UCSF laboratory, and I had the pleasure of meeting him in an international conference. And he works on a lot of viruses, and calls himself the disease detective. In a matter of 3 days, he identified a novel coronavirus in ball pythons, which were dying of pneumonia. He did an autopsy, being a vet, picked up the tissue, and went for sequencing and proved that it's a novel coronavirus. So he's really a disease detective, very wonderful person to interact and work with. And of course, Vinod already told us that the SARS-CoV virus was discovered in a matter of 5 days by the Chinese by direct pregnancy. So I'll share with you some of the experiences that we've had using genomic sequencing much before we had this updated fast versions. These were very laborious ones. We had a collaboration with the University of Liverpool, and this work I did partly in Liverpool and partly in India. We investigated outbreaks of encephalitis that were occurring around Bellary district, and through sequencing we identified 3 viruses. Chikungunya is the first global proof of virus directly in the CSF, implicating its role in encephalitis. The first report of enterovirus 75 causing encephalitis in children, and of course parvovirus 4, all through metagenomic sequencing. We had extensive field experience much before COVID. We had a CDC funded project for 5 years on the etiology of acute encephalitis syndrome in 3 large states of India, Uttar Pradesh, Bengal, and Assam. And this is published in Lancet Global Health. It's the world's largest AES surveillance that has ever been conducted, 14,000 patients. And I think I've spoken about it before. We had an algorithm where we did serological tests and we did molecular tests in the CSF for a dozen pathogens. This algorithm increased the yield of diagnosis from 16% to 50%, but still 50% were unknown. That's the time I had gone along with my colleagues to CDC for training in genome sequencing in 2015. When we came back, all those samples that were negative using conventional methods, we tried to enrich and do sequencing in the CSIR samples. And we identified a large number of ones. One of the first things we identified was parvovirus B19, and this is the first report from Asia on parvovirus B19 causing encephalitis in children. And these were about 13 children from West Bengal. We did the whole genome sequencing, and we were able to show that it was genotype 3B that we found, and it formed a separate layer within the global sequences. We also identified a large number of known pathogens which were not picked up by conventional techniques, like Japanese encephalitis, scrub typhus, dengue, leptospira, chick, strep pneumonia, and sometimes multiple pathogens. 2 pathogens sequences were seen in these patients. This is the sequencing we did at the Assam Medical College Hospital in Dibrugarh, and you can see my team there, and in the lower left panel you can see Dr. Chitra. She was leading the sequencing efforts. The reason why we did sequencing in Assam is, by the time you transport sample to Bangalore, it's late, and CSF is a very precious sample. But we had a very specific question that we were asking. The question was, Assam reports the highest number of Japanese encephalitis. If you look at Assam health records, immunization coverage is over 90%, but still they get a large number of encephalitis. The question we hypothesized was, is it vaccine failure? And we used the Chinese SA1442 vaccine. And there were reports in 2011 and 12 that the SA1442 virus does not afford protection against genotype 5. Genotypes circulating in India are predominantly genotype 3. We have had a couple of genotype 1s, but almost all of them are invariably genotype 3. But southern China, bordering Assam, had reported genotype 5 cases. You know, viruses don't have geographical boundaries, and I hypothesized, could these be vaccine failures because of genotype 5? And we were able to obtain JEB sequences from the CSF. Fortunately, all of them turned out to be genotype 3. The reason why I'm telling you is, it's important to ask the right question. Genomics is a very powerful technology. You can get lost and get fascinated. But if you go with a targeted question, then you can this of course not work from our lab, but very exciting work happening in the last 7-8 years. We all know that human endogenous retroviruses are present in each one of us. During evolution, from the monkeys to man, you know, retroviruses got integrated into human genome. 8% of human genome contains bits and pieces of endogenous retroviruses. Now there is enough evidence to say, due to reactivation, these endogenous retroviruses, through a variety of mechanisms, are contributing to 2 diseases which we did not know the precise etiology, and one is multiple sclerosis and the other is amyotrophic lateral sclerosis. We had the privilege of also participating in the COVID pandemic. We were one of the first founder 10 labs in genomic sequencing, and I was heading the genomic sequencing efforts in Karnataka. And as you can see, the previous speakers have already said it is useful in tracking transmissions, detecting mutations, identifying mutations that affect vaccine potency, and also using the genomic information for development of diagnostics. And while at Tata MD, Dr. Shekhar Pandey is sitting here, IGIB colleagues are here. IGIB had developed CRISPR-Cas9 based COVID-19 detection assay. This was the first CRISPR diagnostic assay developed in the world, one of the first. And they gave it to Tata MD, which commercialized it as the world's first kit. And this transfer and transition from a bench test into a product happened in a record time of 100 days. Over 3.5 million tests were manufactured and sold. It was deployed in the Kumbh Mela, and the product ultimately had to be stopped for production because RT-PCR prices crashed to less than 50 rupees. But the CRISPR test commercially, the cost itself was more than 350 to 400 rupees. So it did not sustain the market. This, of course, is something that I did at Tata MD in the last month of December 2021. As you all know, November 26th, WHO declared the emergence of a new variant called Omicron. 1st December, my boss Girish Krishnamurthy and I had a chat and said, shall we develop a variant detection PCR? Why a variant detection PCR? Because sequencing is costly and relatively more time consuming. I know Vinod used to do it in 48 hours, but most labs had to pull samples. It used to take 5 to 7 days. And so we designed primers by 7. We ordered them, got them. We created a synthetic sample using sequences. December 23rd, we found that it is working. Filed a patent, submitted to ICMR. 31st, ICMR did rapid assessment comparing to genomic sequencing. 1st January, I felt sick and I was diagnosed with Omicron, possibly, but my sample was taken to the lab and tested and turned out to be Omicron positive. And 4th January, we got the CDCO license and went to manufacture. All was hunky-dory. We started, selling the kit, 100% sensitive, 99% specific. ICMR report in comparison to genomic sequencing. 3 weeks later, we get a jolt saying that it's missing some variants. We had created in Tata MD variant watch platform where we continuously monitor in silico whether our primers and probes are going to be effective with the emergence of every new variant. So we calibrated the kit in 4 days and put it back, and touch wood till now, all the emerged variants after that, this kit is picking up. Like in Maharashtra, we also did genomic sequencing to learn some epidemiological lessons. It was lockdown time. We had already started sequencing. Nothing much to do. Routine work had come to a standstill. So we did this sequencing of samples and then showed how the virus entered into Karnataka. It was imported. Large number of the sequences were similar to the Iran and the Italian sequences. And then of course they started circulating. We showed that once it emerged into Karnataka state, it spread wide. So this is something very, very valuable, as Dr. Karyakarte said. But something more interesting, which is very passionate to me, is this paper which we published in Lancet, E-Clinical Medicine. Just data mining, looking at sequences, looking at epidemiology, and that rangoli you see on the right is actually cluster analysis. You see red dots and yellow dots, and they're connected. The cluster analysis tells you that if it is a red dot, it's a symptomatic patient. If the center is yellow, that means it's an asymptomatic patient. Even common sense requires evidence. And the common sense here was symptomatic patients spread the infection to far more number of people compared to asymptomatic infections. And you can see in the outer periphery, large number of clusters, including the largest cluster we had was a pharmaceutical company in Mysore, near Nanjangud, where the sequences, through genomic sequencing, we showed that this index patient was a symptomatic patient and how many people. So in summary then, it's a powerful technology. Use it with a purpose to answer specific questions. Do not be a slave to technology, but recognize its limitations. Expanding metagenic sequencing platforms to improve pathogen discovery and outbreak prediction will be the future. leveraging AI and big data to integrate genomic trends, which both Dr. Pandey and Dr. Scaria talked beautifully, strengthening collaborations between academia, diagnostic manufacturers, and policy makers will help proactive healthcare solutions. Together, we can turn genomic insights into life-saving interventions. A large number of people, my team, they do all the work and I do all the talking. Dr. Chitra, Dr. Anita, my colleagues in Katanavi, Satya, Sunil, and Azar, I want to thank all of them. Thank you.
Thank you, sir. I now call upon our next speaker, Dr. Pandey. Sir is a distinguished structural and computational biologist. He served as the Director General of Council of Scientific and Industrial Research. Among his numerous accolades, he received India's highest scientific honor in the biological sciences category in 2005, that is the Shanti Swarup Bhatnagar Prize for Science and Technology. Currently, sir is a distinguished professor at Bioinformatics Center, Savitribai Pune University, and is an honorary distinguished scientist at the National Center for Cell Sciences, Pune. Sir.
Thank you very much. First of all, thank you, Dr. Karyakarte, for this invitation to speak at this conference. Just like what I said the other day, I am not a medical microbiologist by a long shot. I am not even a microbiologist by a long shot. I just happen to work in a few institutes where I got some exposure, and whatever I've learned is through my friends, colleagues, and conferences like this. Now, since we are talking about genomic analysis in diseases, particularly infectious diseases, I thought I will elaborate a little bit about what happened during the pandemic and how we started working on the genomics aspect of the pandemic. As earlier speakers have already said, that because we are in the modern era in which we have all these tools available, it was possible to very rapidly detect the cause of the disease as that due to SARS-CoV-2, Vinod has already said. If you recall, sometimes in the last week of December 2019, there were a cluster of pneumonia-like cases that were reported from the Wuhan province of China. And a cluster of pneumonia-like cases could have been due to anything. But very soon, doctors were able to isolate the samples from the lungs and others, from sputum, and they were able to sequence the first genome of this particular cause. It was not known whether it was bacteria, fungus, or virus, but the sequencing revealed something like 7th January 2020. It revealed that the sequence is very similar to coronaviruses. In fact, it was very similar to, it appeared to be a cousin of SARS-CoV-1, and therefore it was actually called SARS-CoV-2. So that's what it was done. The CDC Atlanta announced the first genome sequence on 10th January 2020. First case in US was reported sometime in mid-January 2020. In India, we reported our first case in Kerala on 30th January 2020. So that's how the disease actually started. By then, of course, it had reached Europe. One of the first countries it had reached was Italy, and that was because of the very strong trade relationship between Wuhan province of China and Italian textile industry. So that's how the disease actually started spreading. I had an opportunity to come to Pune on 7th February 2020, just 7 days after the first case was reported. And I was giving a talk at the Bengaluru auditorium. And I did mention in my talk that we are probably living in the coronavirus era.
So it was not yet identified that it was going to be such a serious pandemic, but we already had been picked by the government that one has to actually watch out where we are going. In CSIR, we had a meeting of all the directors of CSIR labs. There were 47 different labs. Rajesh was there in that particular meeting. And that happened on 24th, 25th February 2020 in Goa. And we discussed there what would be our strategy if such a reality were to dawn upon us. Of course, we had some very brilliant people with us. Someone like Vinod Agarwal, who was also a director of IGIB. And at that time, we sat together to discuss our strategy. And like the other day I said, we decided that at least in CSIR, we have a 5-pronged strategy. The first arm would be surveillance. The second arm would be diagnostics. The third one would be interventions. The fourth would be hospital-assisted devices. And the fifth would be supply chain logistics. And because the surveillance was one of the very important arms that we decided we were going to work on, mind you, by then, the WHO had still not called this as a global emergency. WHO declared a global pandemic emergency only on 11th March 2020. So it's full 15 days before that we already had our strategy in place. And we actually discussed this with the government, we discussed this with ICMR, we discussed it with CDC. I had long discussions with PMO themselves that should an emergency come, what do we do? And then it was decided that the first thing that we were not prepared, we would do RT-PCRs in the country. That we would have a lockdown. That was actually a decision that was very consciously taken in government that we would have a lockdown as soon as possible. And then 25th March, actually the government declared a national lockdown at that particular stage. The lockdown was essentially, on one hand, to stop the spread of the disease, on the other, to prepare ourselves on various things. For example, CCMB, Rajesh will tell you, trained a large number of people from hospitals in collecting samples and doing RT-PCRs. RT-PCRs were not available in most of the hospitals. And in fact, people in Nizam's hospitals and all, they actually came to CCMB and we trained all of them in different institutes. In Calcutta, for example, the Calcutta Medical College did not know how to do RT-PCR. And RB College in Calcutta, we donated our 2 RT-PCR machines to them so that they could actually get in place to start doing RT-PCR. So this all happened during the lockdown. States like Ladakh did not have any infrastructure whatsoever. So we airlifted 2 RT-PCR machines from Jammu and went and placed them in Ladakh. So that's how actually we literally spread all over. And ICMR and CSIR, we were working hand in hand together on at least enhancing our capability of detecting and reporting to NCDC. So NCDC was the center point where it would actually be reported. And very soon, we discussed, so in CSIR we had a strategy group, which used to meet every evening at 6 o'clock, including Saturdays and Sundays, online. And we had discussed that genomic surveillance would be a backbone of that particular strategy. And in terms of surveillance, we added 3 more arms to surveillance. Genomics was the first one that we began with, and of course IGIB and CCMB took lead that, and both Rajesh and Vinod are here. And Vinod has already told us about it. Rajesh also will tell a little bit about sequencing, how we actually started. And at that particular time, I mentioned very briefly the other day, we had some resistance from some department of the government, the one who was actually kind of taking care of the pandemic response, saying that NIV had sequenced the first virus isolate, and we don't think that we need to sequence another. I recall we had a meeting in Honorable Health Minister's office. I was present there myself, and I strongly pitched saying that if this particular agency of the government does not want to do sequencing, we will do it upon ourselves. And that's how we began doing sequencing in CSIR. And CCMB and IGP, of course, took the lead and started sequencing from a variety of samples across the country. So that was the one
Focus
of our surveillance strategy. But we added 3 more. One, we also started doing zero surveillance. And that would actually tell us that how fast the disease was spreading across population. So zero surveillance was at multiple point of times, before vaccination, after vaccination, and so on and so forth. We also added wastewater surveillance, And Pune, of course, and also Dr. Karyakarte also was a part of that, did a fantastic job in wastewater surveillance and did give us, as I said, about 15 days advance notice of the spread. And the fourth arm that we added was aerosurveillance. The other day I spoke about it. Because the disease is airborne, we did that. We had a program, Vinod has already mentioned about it, with the state government of Kerala, where every week we were receiving some samples from them and we were doing sequencing. And that helped us in actually tracking the spread of the virus through Kerala. And a similar program was set in
Wakad.
with Maharashtra government sometimes in 2021, I believe around April or something like that. I had a personal conversation with the then Chief Minister of Maharashtra myself, and we decided that in Maharashtra also from every district there will be samples collected and we would do sequencing. And at that time, I was helping quite a lot in sequencing. And Dr. Karyakarte took the lead in characterizing the clinical data.
लेकिन गाड़ी नहीं है ना जाएगा कहीं
So these are the kind of surveillance strategies that what we had. We decided at a time and we put it there. Dr. Karyakarte already mentioned in his talk the fruits of that. That was extraordinarily important for us to have done that. Over the time, after the first two or the third wave, the severity of the virus thankfully came down, and I was talking to some of the students the other day, that every disease, or rather every viral disease, has a very characteristic first 2 or 3 waves which are very deadly, but as successive waves come, they become lower and lower in the intensity of their pathogenicity or whatever. Perhaps because of the immunity that is set in, or the people have already antibodies against the virus with the subsequent wave and so on and so forth. And it has happened in multiple earlier disease outbreaks, like for example influenza and so on and so forth. And SARS-CoV-2 is no different in that particular respect. But yet, it is still important to be vigilant. And for the future pandemics, now that we have capability, I don't think we should lose this particular capability of surveillance like what we have done. And as I said, surveillance of all respects, genomic, sero, and wastewater, all of them are extremely important from the public health perspective. And as world is looking at, for example, issues like AMR, all of these actually become important in this surveillance aspect. And I do hope, like the success that we had in Maharashtra, we can similarly replicate that all over India in putting in place a very strong surveillance network. And in the coming time, if we can actually convince NCDC or the government of India of having specific surveillance strategies which continue through the time and help us in identifying any new outbreaks or new disease outbreaks, we will be well prepared much before time than the last time. So thank you all very much.
Thank you, sir. Our next speaker, Dr. Subhash Salonke. Sir started his public health career from 1973 with over 50 years of experience in public health department, from deputy director to the only director general in health services of Maharashtra state. He also serves as the WHO country head in Indonesia and assistant regional director at WHO for Southeast Asia region. Sir will be sharing his perspectives and his role in public health leadership, collaboration with microbiology at state and national level.
Thank you very much. I'm sure you are finding this session getting a little delayed. I won't take more than 10 minutes. Rajesh, thank you very much. And you have given me an opportunity to interact with the stalwarts like Dr. Pandey and others, and also the colleagues here. I wish to only indicate to you a kind of an historical perspective, that has been indicated that since 1976, nearly 50 years, of public health scenario in this country. I'm part of that. Smallpox, last year's measles outbreaks. I was involved in different levels. I have seen the microbiologists, medical microbiology and the government background and not being considered as something very important in those days. Now you are heroes. We can't think of doing anything as far as surveillance is concerned, as far as management of the patients are concerned without you. There is no question of that. And the progress that has gone in, particularly in the last few years, thanks to the COVID did a great thing. Obviously, COVID has taught us a lot of things, and we only hope that we don't forget them. Unfortunately, we have a great tendency of forgetting very bad things in a very short span of time, and also forget that capacity building exercise, which need to be continued continuously, and I think also essential that the coordination and the collaborative efforts. Yesterday, there was a wonderful presentation by colleagues both from ICMR about One Health.
त्याच्यामुळे नाही होते ते स्क्रीनमुळे होते
Involvement of the public health approach that has gone in since at least from 1976 till now. We have seen that during COVID, actually every country has made mistakes. We have made mistakes. There are some people who will say that India did a great job. Yes, we have. But we have also done something which we could have avoided not many cases and also deaths. One of the classic examples, as Rajesh has already indicated, about the Amravati kind of a situation. I distinctly remember, the cases from Amravati district were being reported in a large number. And I was a part of the both national task force. I still continue to be, I don't know, because that particular ICMR task force has not disbanded. So when it is not disbanded, it seems to be there. It is in the kind of an जिंदा भी है मर भी गया है कुछ भी बात नहीं है but that task force is there. So from that task force in the state level task force, advisor to the CM, advisor to the deputy CM, then he requested me, he said, Dr. Sanke, you have to go to Amravati, and I had been to Amravati as I just stated. I went right up